Is proton radiation more effective than photon radiation at inducing senescence? Background: breast cancer is the most common cancer in women in US. It's also the second deadliest. BRCA1 and BRCA2 are genes that help fix damaged DNA. This stops cancer from forming, but if these genes have harmful changes, they can cause cancer. Everyone gets one copy of each gene from each parent. These changes can also be passed down. We are looking at two types of human breast cancer cells. One type has changes in BRCA genes. The other type has normal BRCA genes. We are also looking at a type of mouse breast cancer cell. Senescence is when a cell stops dividing forever. This can happen because a cell has divided too many times, or it can happen because something stressful has happened to the cell, like radiation. Radiotherapy uses radiation to damage DNA. This can make cells die or stop dividing. Methods: the paper uses a marker called SA-beta-gal to find and count senescent cells. There are two ways to do this. The first way is a histochemical assay. Cells are put on glass cover slip in 6 well plates. The cells are then hit with 10 centigrade protons and photons when they are not too crowded. The cells are left for 7 days to become senescent. Then they are stained with the invitrogen beta-gal staining kit. This kit uses X-gal, which is substrate of SA-beta-gal. When X-gal is cleaved, it makes the blue compound that can dissolve. Senescent cells can be found by the looking for blue spot. The second way is flow cytometry. Cells are put in T12.5 flask. The cells are then hit with 5 centigrade protons and photons, or treated with H2O2 when they are not too crowded. The cells are left for 7 days to become senescent. Then they are prepared for flow cytometry. This involves a 1-hour incubation with bapillomycin A1 to remove background senescence. Then there is a 2-hour incubation with C12FDG. C12FDG is a substrate of SA-beta-gal that glows when it cleaved. The samples are then run in the flow cytometer. Flow cytometry results. Figure 1 shows plots for 4 T1 cells that were not irradiated. The first graph or graph A separates the cells from debris. The second graph or graph B selects only the living 4 T1 cells. In the third graph or graph C, the top 2 sections contain senescent cells. For the non-irradiated samples, senescent is set 2% and irradiated samples are analyzed relative to the non-irradiated samples. Figure 2 shows plots of 4 T1 non-irradiated samples in red overlaid with 5 centigrade X-rays and 5 centigrade protons in blue. Each plot represents one independent experiment for at least 5 times 105 living cells. Figure 3 shows the percent of senescent cells at A, B and C varying radiation types and doses. Each bar represents the mean of independent experiments. X-gal staining results. Figure 4 shows images from X-gal staining. The small dark spots clustered within cells are insoluble blue compounds released by X-gal, and the cells that they are clustered within are senescent cells. Images are of 4 T1 irradiated with 0 centigrade at A and 10 centigrade at B. Discussion. The paper is working to get clearer X-gal staining images. They are using glass cover slips and fewer cells for this. More experiments are being done right now. Based on the results so far, it seems that protons cause most senescence in 4 T1 cells, but X-rays cause most senescence in HCC 1937 BRCA cells. The paper thought that protons would cause most senescence because they are bigger than photons and have a higher linear energy transfer (LET). This means they cause more double-strand breaks and more stress on the cell. After they finish their in vitro experiments, they plan to do in vivo experiments. They hope that the in vitro results will be in the same in vivo and more senescence will lead to better control of the tumor.